Welcome to the Deep Dive. Today we're embarking on an exploration that drills down to the very foundations of animal life. You might think of animals in terms of complex behaviors or, grand ecosystems, but what we're about to unpack shows how every spectacular function and even their surprising demise hinges on the tiny, intricate world of molecules and cells.
Precisely. Our source material, a chapter from Hill, Wise, and Anderson's Animal Physiology, it kicks off with some truly remarkable examples. Imagine a slow-moving predator conquering fast-moving prey. It sounds, well, wrong, doesn't it?
Yeah, completely counterintuitive.
Consider the almost motionless cone snail preying on a fish, or the notoriously sluggish puff adder taking down a nimble rat. What's their secret? It's all about molecular level disruption.
Okay, so it's not speed, it's sabotage on a tiny scale.
Exactly. Both the puff adder and the cone snail unleash venoms that specifically attack and dismantle vital molecular structures in their prey. The puff adder, for instance, needs only a second or two of contact. Its venom, it's this complex cocktail of compounds, basically strips membranes from muscle cells and pokes holes in blood capillaries,
Wow.
leading to widespread internal hemorrhaging. The rat's molecular and cellular mechanisms simply fall apart, paralysis, and then, well, consumption.
Grim, but effective.
Uh,
And the cone snail.
The cone snail takes it to another level almost. It uses a worm-like body part, like a lure, to tempt the fish closer.
Clever.
Once a fish gets close, the snail spears it and injects compounds that almost instantly disrupt proteins essential for the fish's nerve and muscle cells. Its most promising defense, the ability to swim rapidly away, is immediately defeated. Gone.
So the escape plan is just neutralized at the source.
Right. And this powerfully reminds us that all higher functions of animals, from a racehorse's sprint to the intricate thoughts of a mind like Socrates, utterly depend on the proper function and organization of molecules and cells.
So for our deep dive today, our mission is clear. We're going to explore these fundamental structural and functional properties. Molecules and cells, that's where the action is. We'll peel back the layers on cell membranes, epithelia, the incredible world of enzymes, and how cells receive and act on signals. You'll see how comparative strategies and experimental methods illuminate adaptive significance, and we'll pepper in some incredible real-world examples to really bring these concepts to life for you. The molecular foundations, membranes, and cells.
Okay, let's start at the absolute basics. The cell membrane. Every single animal cell is encased in this thin barrier, only about 6 to 8 nanometers thick.
Nanometers, yeah, incredibly thin.
And within the cell, countless intracellular membranes define internal compartments. But these aren't just, inert walls. They're dynamic architects that actively compartmentalize systems. They allow the inside of a cell to have vastly different properties from its outside, and they're also crucial for receiving and transmitting signals, making them far more than simple boundaries.
And the very core structure of these membranes is, well, it's a masterpiece of design, isn't it? It's primarily a double layer of phospholipid molecules
That's the key part.
with various proteins embedded within. We often think of it as a fluid mosaic, constantly in motion. What makes that fluid mosaic truly work? What's the principle?
It comes down to molecular polarity. You know how oil and water don't mix, right?
Sure, cell dressing 101.
Exactly. Oil is nonpolar, or hydrophobic, literally water-fearing, while water is polar, or hydrophilic, water-loving. Phospholipids are fascinating because they're empathic, meaning they have both parts, a polar, water-loving head and a nonpolar, water-fearing tail.
So like a molecule with two personalities.
Sort of. And this dual nature is why they spontaneously form bilayers in water. The hydrophobic tails tuck into the core, away from the water, while the hydrophilic heads face the water on either side. It just happens.
Okay, so it's not just a static structure. These phospholipids are incredibly diverse. You mentioned human red blood cells have over 150 different kinds.
That's right. Huge diversity.
And they're constantly moving, able to diffuse around the entire circumference of a cell in minutes. The key insight here seems to be how subtle molecular changes allow life to thrive in really extreme environments.
That fluidity is absolutely critical, and it adapts. It adapts to environmental factors. For example, the degree of chemical saturation in the phospholipid tails affects it.
Saturation, like in fats.
Precisely. Double bonds create bends, or kinks, in the hydrocarbon chains. This prevents tight packing and increases fluidity. And this is a brilliant adaptive strategy you see in action. Polar fish, living in extremely cold water, have evolved cell membranes rich in these double bonds.
To stop them freezing solid, basically, or getting too stiff.
Exactly, keeps their membranes from becoming detrimentally stiff and dysfunctional at low temperatures. Even individual animals, like lab mice before hibernation, can dynamically restructure their membrane phospholipids to prepare for the cold. They change their own chemistry.
That's amazing. And the proteins. The proteins embedded within this fluid mosaic. They're not just passive components, are they?
No, not at all. They endow membranes with huge functional capacities. Our sources identify 5 key functional types that orchestrate pretty much everything that happens at the cell surface.
Okay, what are they?
Channels, transporters, enzymes, receptors, and structural proteins.
Right.
Channels are like tiny gates, creating pores for specific things like ions or water. Transporters act like ferries, binding to and moving molecules across.
Right.
Enzymes embedded in the membrane catalyze reactions right there on the spot. Receptors are the cell's antenna, binding with specific signals from outside to initiate changes inside.
Structural.
Structural proteins provide an anchor, linking the membrane to other cell parts or forming junctions with neighboring cells. And what's truly remarkable is that a single protein can often wear multiple hats, perform several functions. Membranes are incredibly versatile.
So the cell membrane is this dynamic adaptive barrier, teeming with specialized proteins all working together. It's a marvel of molecular engineering, controlling what comes in and out. But cells rarely work alone, do they? Building on that idea of intricate boundaries, let's zoom out a bit and see how these individual cells organize themselves into larger protective sheets called epithelia. Building blocks of bodies, epithelia.
Epithelia, yeah, they're essentially like the body's internal and external skin, forming these crucial boundaries. A simple epithelium, for instance, is just a single layer of cells. You find them lining your intestines, kidney tubules, blood vessels, lots of places.
Okay.
Each cell has an apical surface facing a cavity or the outside world, and a basal surface attached to the underlying tissue, usually resting on a thin basement membrane.
And these aren't just flat sheets like tiles on a floor.
No, their shape varies a lot depending on the job. They can be squamous, low and flat, or cuboidal, like little cubes, tall as they are wide, or columnar tall and thin.
Form follows function again.
Always. And many also feature microvilli, these incredibly fine, finger-like projections on the apical surface, giving them a brush border appearance under the microscope.
Right, I've heard of the brush border. What's the advantage?
Microvillia are a classic example of adaptive morphology, perfect illustration of form and function. In the intestinal epithelium, for instance, they vastly increase the surface area.
For absorption.
Exactly, for absorbing and digesting materials much more efficiently. You see them in any epithelia active in intense secretion or absorption, like certain kidney tubules too. It's a simple structural change, just folding the membrane, but with a profound physiological impact.
What's especially intriguing is how these epithelial cells connect to each other. They form these incredibly tight barriers sometimes, or allow for very specific communication. Our sources mention 4 key types.
That's right, 4 main types of cell junctions. Tight junctions, septate junctions, similar function mainly in invertebrates, desmosomes, and gap junctions.
Let's talk tight junctions. They sound important.
They are foundational, really. They're where the cell membranes of adjacent cells essentially fuse, completely blocking the intercellular space. Imagine a continuous ring, like a zipper, around each epithelial cell. This creates a fence that separates the apical surface from the basolateral surface, that's the basal and side surfaces combined.
Okay, so it divides the cell surface into 2 distinct regions.
Precisely. And this means the 2 regions of the cell membrane can have different proteins and different functions. This leads to what's called functional asymmetry. And this asymmetry is absolutely vital for directed transport across the epithelium.
So this functional asymmetry is key to how epithelia control transport. Substances can pass through the cells via transcellular paths or between them via paracellular paths. And the tight junctions are the gatekeepers for that between route.
You got it. Tight junctions regulate that paracellular movement. Some epithelia have leaky tight junctions, allowing more movement between cells, like in parts of your kidney where lots of water needs to move.
Right.
While others, like the blood-brain barrier, have incredibly tight junctions that almost completely prevent leakage.
Makes sense.
For transcellular transport, understanding the distinct proteins and functions of both the apical and basolateral membranes is essential. Things go in one side and out the other specifically.
What about the other junctions? Desmosomes and gap junctions.
Desmosomes act like spot welds or rivets. They provide mechanical strength, holding cells together against physical stress, like in your skin. Gap junctions are different. They create open pores, little channels, directly connecting the cytoplasm of adjacent cells. This allows small molecules and ions to pass directly between cells, vital for rapid cell-to-cell communication, like coordinating heart muscle contractions.
Okay, so membranes create boundaries within cells, and epithelia create boundaries between body compartments using these specialized junctions. Now let's pivot to the powerhouses behind all cellular chemistry.
Here.
Enzymes, the engine of life enzymes.
Enzyme.
Okay, these are the protein catalysts, right? They're the workhorses, but also the managers.
That's a good way to put it. They're the master conductors of life's chemical symphony. They play two enormous roles. They accelerate chemical reactions by incredible magnitudes, sometimes millions or billions of times faster.
Wow.
And they meticulously regulate them, ensuring everything happens in the right place, at the right time, at the right speed.
Can you give an example of how critical that speed is?
Sure. Think about Mark Twain's celebrated jumping frog, Dan Webster. Why could a leopard frog jump so much faster than, say, a toad?
Better muscles.
Partly, but it's not just muscle fiber differences. A key reason is that frogs are much better endowed with a specific enzyme, lactate dehydrogenase, or LDH. This LDH allows them to make ATP, cellular energy, much faster without oxygen, which is crucial for those lightning-fast escapes from predators. The enzyme itself is often the limiting factor for burst activity.
So enzymes aren't just accelerators. They essentially determine which reactions even happen at a significant rate under the conditions inside a cell.
Exactly. They orchestrate a cell's entire biochemistry, making life as we know it possible. Without them, most reactions would be far too slow.
How do they actually work? What's the mechanism?
It's elegant, really. An enzyme must first bind with its substrate, that's the molecule it acts upon, to form an enzyme-substrate complex.
Okay.
This binding is usually temporary, stabilized by weak, noncovalent bonds. The enzyme then lowers the activation energy required for the substrate to transition into product. Think of it like lowering the hump a ball needs to roll over to get down a hill.
Makes it easier for the reaction to happen.
Much easier. And crucially, the enzyme itself emerges unaltered at the end, ready to catalyze another reaction, sometimes thousands or even millions of times per second. This catalytic effectiveness, the sheer speed measured by the turnover number, is just astonishing.
That speed is incredible. And enzymes also exhibit what's called saturation kinetics. Can you explain that?
Yeah, think of it like checkout lines at a store. If there's only one cashier enzyme and few customers, substrate, adding more customers speeds things up.
Right.
But eventually, the cashier is working as fast as possible. Adding more customers now just makes the line longer. The rate of checkout, reaction velocity doesn't increase further. The enzyme is saturated.
So there's a maximum speed, Vmax.
Exactly. And this Vmax depends on how much enzyme is present and how fast each enzyme molecule can work. But there's another key factor. Enzyme substrate affinity, measured by the Michaelis constant.
Affinity. How strongly they bind.
Right. A low CalM means high affinity. The enzyme binds the substrate tightly, even at low concentrations. A high CalM means low affinity. Since substrate concentrations in cells are often below saturation levels, this affinity is vital in determining the actual reaction speeds happening inside the cell. It's all about the interplay of enzyme quantity, its maximum speed, and its attraction to its target.
What's also fascinating about enzymes is their flexibility. I remember the old lock and key analogy for how they bind substrate.
Yeah, that's a bit outdated, or at least needs refinement.
Why?
Because the enzyme's active site isn't rigid like a lock, it's flexible. When the substrate binds, the enzyme often changes shape slightly to achieve a better fit. It's called induced fit. Think more like a flexible glove adjusting to a hand rather than a rigid key in a lock.
Ah, okay. That makes more sense, more dynamic.
Much more dynamic. And this flexibility is key to their function and regulation. Many enzymes are also multi-subunit, meaning they consist of two or more protein chains bonded together non-covalently.
And that matters
Because these often exhibit cooperativity. Binding a substrate molecule at one active site can change the shape of the other subunits, influencing how easily they bind their substrates.
like teamwork within the enzyme molecule itself.
Exactly. Binding at one site can cause a ripple effect through the whole molecule, making subsequent binding easier or harder. This allows for really intricate, rapid regulation.
Which brings us to allosteric modulation.
Yeah.
That sounds like a key regulatory mechanism.
It is. It's super important. Here, molecules that are not the substrate, called allosteric modulators, bind to specific regulatory sites on the enzyme away from the active site.
Sure.
This binding changes the enzyme's shape, altering its catalytic activity, either increasing it, activation, or decreasing it, inhibition. It's almost an instantaneous switch governed by the concentration of the modulator. Allows cells to instantly ramp up or slow down entire metabolic pathways based on current needs.
Can you give an example of that?
A classic one is phosphofructokinase, or PFK. It's a pivotal regulatory enzyme in glycolysis, the pathway that breaks down sugar for energy. Its activity is inhibited by high levels of citrate. Citrate is a product of later steps in energy metabolism, so high citrate signals the cell has plenty of energy, no need to break down more sugar right now.
Makes sense. Feedback.
Right, it's feedback inhibition.
Mm-hmm.
And conversely, PFK is powerfully upregulated, switched on, by high levels of AMP. AMP builds up when ATP, the main energy currency, is low. So high AMP signals an energy crisis, telling PFK to kick glycolysis into high gear. It maintains perfect metabolic balance.
Ingenious. Beyond this allosteric way, there's also covalent modulation.
Yes, another major regulatory strategy. This often involves phosphorylation, the addition or removal of phosphate groups to the enzyme protein itself.
Like attaching a little chemical switch.
Precisely. Specific enzymes called protein kinases add the phosphate groups, often turning the target enzyme on, while protein phosphatases remove them, turning it off. It's a very rapid on-off switch.
And this can lead to amplification.
Oh, huge amplification. Imagine a domino effect. A single activated signaling molecule might activate one protein kinase. But that one kinase molecule can phosphorylate and activate many molecules of a second protein kinase.
Okay.
And each of those can activate many molecules of the target enzyme. This cascade means a tiny initial signal, like a single hormone molecule binding to a receptor, can trigger a massive cellular response down the line. It's incredibly efficient signal boosting.
Finally, it's worth noting that enzymes aren't identical everywhere, right? Even for the same job.
Correct. Our sources explain isozymes. These are different molecular forms of the same enzyme within a single species, like the LDH enzyme we mentioned. Humans have different isozymes, LDHA4 dominant in skeletal muscle, good for anaerobic bursts,
Yes.
and LDHB4 dominant in heart muscle, better for steady aerobic work. Same basic function, but fine-tuned for different metabolic needs.
And between species.
Between species, we talk about interspecific enzyme homologs. These are similar enzymes in different species that have evolved slightly different properties. For example, the LDHA4 enzyme in rockfish living in deep, cold, high-pressure water has adapted to function better under those extreme conditions compared to the LDHA4 in their shallow water relatives. It's a testament to the adaptive power of evolution tweaking these molecular machines.
Okay, so enzymes are regulated, adaptable, and absolutely essential. The dynamic duo protein life cycle and signaling.
Absolutely. And given how vital proteins and enzymes are, it's probably not surprising that cells have really sophisticated dynamic mechanisms for their entire life cycle, not just making them, but maintaining them and getting rid of them too.
Right, because things can go wrong.
Things can definitely go wrong. Proteins can be denatured, lose their proper three-dimensional shape due to stresses like extreme heat, changes in pH, or toxins, and a denatured protein is usually a useless protein.
So what do cells do? Just make new ones?
They can, but they also have repair crews. These amazing molecular chaperones, like the famous heat shock proteins, you might see them abbreviated as HSP70, HSP90.
Heat shock proteins.
Okay. They act like cellular first responders. They can bind to damaged or misfolded proteins and, using energy from ATP, help guide them back to their correct functional conformation. We see this in action with mussels and snails on rocky shores. When they get baked by the sun at low tide, they ramp up production of these heat shock proteins to repair the heat damage to their cellular machinery. It's a crucial survival mechanism.
Wow, cellular repair kits. But what if the damage is too severe, or the protein just isn't needed anymore?
Good question. When proteins are beyond repair or simply past their use-by date, cells have a highly specific, elegant degradation system, the ubiquitin proteasome system.
Ubiquitin sounds ominous.
It's sometimes called the kiss of death for proteins. Small ubiquitin molecules get attached to the target protein, like tagging it for disposal. Multiple ubiquitins form a chain.
A molecular tag.
Exactly. This tag is then recognized by a large protein complex called a proteasome. The proteasome acts like a miniature recycling plant, or, maybe more accurately, a shredder. It unfolds the tagged protein and chops it up into small peptides.
And then?
Those peptides are further broken down into individual amino acids, which can then be reused to build new proteins or oxidized for energy. It's a wonderfully efficient and highly regulated recycling program. No junk building up.
Okay, so cells maintain their protein workforce carefully. Now that we've glimpsed this incredible molecular work, let's zoom out and see how these mechanisms translate into some truly spectacular, visible adaptations in the animal kingdom, like light.
Yes, bioluminescence, the biochemical production of light by living organisms. It's surprisingly widespread, especially in the deep ocean where there's no sunlight. And get this, it's evolved independently over 40 times.
40 times, that suggests it's really useful.
Hugely useful. Animals use it for everything, attracting mates, luring prey, camouflage, startling predators, you name it.
How does it work chemically?
Typically, it's a luciferin-luciferase reaction. You have a light-emitting compound, the luciferin, which is oxidized by an enzyme, the luciferase. This reaction releases energy in the form of light, and sometimes you see further modifications, like in the Aquaria jellyfish. They first produce blue light through a luciferase reaction, but then another protein, the famous green fluorescent protein, or GFP...
GFP, used in labs all the time now.
Exactly. That GFP absorbs the blue light and re-emits it as green light, so the jellyfish glows green. It shows how different light-producing or modifying components can combine for a final visual effect. A natural light show powered by enzymes and specialized proteins.
Yeah, and perhaps the most captivating example involves squid.
Ah, yes, the Hawaiian bobtail squid. This is phenomenal. The squid doesn't actually make its own light. Instead, it acquires specific light-emitting bacteria, Vibrio fischeri, from the surrounding seawater.
It farms bacteria.
It essentially farms them in a special light organ, and then it uses the light produced by these bacteria for counter-illumination.
Counter-illuminate.
Yeah. At night, it projects light downwards from its body, adjusting the intensity to precisely match the moonlight filtering down from the surface. This eliminates its shadow when viewed from below by predators hunting upwards.
That is incredible. Hiding in plain sight using borrowed light. A partnership across kingdoms.
It's a stunning example of cross-phylum coordination for survival. Truly amazing.
Okay, from light production to color change. Some animals are masters of camouflage.
Right. Rapid color change, or more accurately, physiological color change, is seen in familiar creatures like frogs, many fish like flatfish, and crustaceans. This often involves specialized cells called chromatophores. These cells are packed with pigment granules, black, brown, red, yellow.
Okay.
The animal changes color not by making new pigment, but by moving these granules around within the cell. They can disperse the granules throughout the cell to show the color, or aggregate them into a tiny spot in the center to hide the color.
How do they move them?
Using motor proteins like tiny molecular trucks that travel along tracks made of microtubules within the cell. It's a relatively slow process, taking seconds to minutes, but very effective for blending in.
But not all color change is slow, right? What about squid and octopuses? They seem almost instantaneous.
Ah, cephalopods. Yes, they are the undisputed champions of rapid color change. Their trick is entirely different and breathtakingly fast. They don't just move pigment granules within a cell, they have tiny, complex color change organs, also sometimes called chromatophores, but structurally different. Each organ consists of a central elastic sac filled with pigment, surrounded by tiny muscles arranged radially, like spokes on a wheel.
Muscles attached to the pigment cell.
Directly attached. When these muscles contract, they pull the pigment sac outwards, stretching it into a large, flat disc, instantly revealing its color over a wide area. When the muscles relax, the elastic sac snaps back into a tiny, almost invisible dot.
Wow. So it's direct muscle action on the pigment itself.
Exactly. And because muscle contraction and relaxation can happen incredibly quickly, milliseconds, they can change color patterns across their entire body almost instantaneously. Flashing, rippling patterns. It's how they communicate, camouflage, startle predators. It's visually stunning.
Okay, so controlling light, controlling color. All this requires coordination. Which brings us finally to cell communication. How do cells talk to each other to orchestrate these complex processes?
Communication is absolutely critical. Everything from muscle contraction to hormone release relies on it. Extracellular signals, things like hormones traveling in the blood or neurotransmitters released at nerve endings, initiate their effects by binding to specific receptor proteins on target cells.
So the signal molecule itself doesn't usually go into the cell, it just knocks on the door.
Often, yes. The receptor protein is the doorbell, and also the mechanism that relays the message inside. This binding causes a precise change in the receptor's shape, its conformation.
And that shape change is the signal.
That shape change is the initial event that sets off a chain reaction inside the cell. Our sources categorize these receptors into 4 main types based on how they work: ligand-gated channels, G-protein-coupled receptors, and enzyme-linked receptors, and intracellular receptors.
Okay, let's break those down. Ligand-gated channels.
These are fascinating because the receptor is the channel. A ligand, that's just a general term for a molecule that binds to a receptor, like the neurotransmitter acetylcholine, binds to the receptor protein, and that binding directly causes the channel part of the protein to open. This allows specific ions, like sodium and potassium in the case of the acetylcholine receptor at the neuromuscular junction, to flow across the membrane, which immediately changes the cell's electrical voltage. It's a very direct and fast signaling mechanism.
Direct link between binding and ion flow.
Exactly. And this type of receptor is a key target for venoms. Remember the cone snail we started with?
Yeah, paralyzing the fish instantly.
Its aconitoxin specifically blocks these acetylcholine receptors on the fish's muscle cells. No signal gets through, muscles can't contract. Paralysis. What's even more striking is that certain crate snakes, venomous snakes, and poison dart frogs have independently evolved remarkably similar toxins targeting this very same receptor type.
Convergent evolution targeting a critical communication link.
Precisely. It highlights just how crucial this single receptor is for basic functions like movement and therefore survival, and what a potent target it makes.
Okay, what about G-protein-coupled receptors? That sounds more complex.
They are a bit more indirect, but incredibly widespread and versatile. In this case, no chemical actually passes through the membrane via the receptor itself. Instead, an extracellular first messenger, like a hormone, say epinephrine or adrenaline, binds to the receptor on the outside.
Okay.
This binding causes the receptor to change shape, which allows it to interact with and activate an intermediary molecule on the inside of the cell membrane called a G protein.
G-protein, like a middleman.
It's exactly like a middleman. The activated G protein then typically moves along the membrane and activates another protein, usually an enzyme. This enzyme then synthesizes an intracellular second messenger, like cyclic AMP.
First messenger outside, second messenger inside.
You got it. It's a relay system. The external signal is transduced into an internal signal.
And you mentioned amplification with these earlier.
Yes. This is where the amplification truly skyrockets. It's a masterclass in cellular efficiency. A single activated receptor molecule can activate many G protein molecules before it gets deactivated.
Okay, step 1, amplification.
Then each activated G protein might activate one molecule of an enzyme like adenylate cyclase. But that single enzyme molecule, once activated, can churn out thousands of second messenger molecules, like cyclic AMP.
Wow, thousands from one.
And it doesn't stop there. Those thousands of cyclic AMP molecules then typically activate protein kinases, leading to that phosphorylation cascade we talked about, where each step multiplies the signal further.
So the signal gets bigger and bigger at each step.
Exponentially bigger. The epinephrine signal in liver cells is the classic jaw-dropping example. A tiny amount of epinephrine binding to receptors on the outside can lead to a 10,000-fold amplification just in the production of cyclic AMP.
10,000 times.
And then the protein kinase cascade triggered by KMP can amplify the signal by another 1,000 times or more, the end result. A potential 10 million-fold increase in the rate of glucose release from stored glycogen, all triggered by a handful of hormone molecules.
That's incredible efficiency. A tiny whisper outside becomes a massive shout inside the cell.
It's how hormones can have such powerful effects even at very low concentrations in the blood.
Amazing. What about the other two receptor types quickly? Enzyme-linked and intracellular.
Right. Enzyme-linked receptors are also involved in signaling cascades. In some cases, the receptor protein itself is an enzyme on its intracellular side, activated by ligand binding. In others, the receptor directly links to and activates an enzyme partner when the ligand binds. They often generate second messengers, like cyclic GMP.
Okay, and intracellular.
Intracellular receptors are different because they're inside the cell, in the cytoplasm or nucleus. They are for signaling molecules that are small and hydrophobic, or lipid-soluble, like steroid hormones, testosterone, estrogen, cortisol, or thyroid hormones.
Because those can pass right through the cell membrane.
Exactly. They don't need a surface receptor. They diffuse through the membrane and bind to their specific receptor protein inside. This activated receptor hormone complex then typically moves into the nucleus and directly binds to DNA, acting as a transcription factor to turn specific genes on or off.
So they directly control gene expression,
Nope.
a slower but more long-lasting effect usually.
Generally, yes. Compared to the rapid changes caused by ion channels or CAMP cascades, changing gene expression takes longer but can lead to more profound, sustained changes in the cell's function or structure. Outro.
So there you have it. A really deep dive into the microscopic world that powers animal physiology, from the incredible flexibility of a cell membrane adapting to freezing cold, to the stunning precision of an enzyme's active site orchestrating metabolism, to the lightning-fast communication that allows for instant color change or escapes from predators.
Or successful predation.
Right. The intricate dance of molecules and cells underpins every single function, every adaptation, every survival strategy in the animal kingdom. It's kind of mind-blowing when you think about it at that scale.
It truly demonstrates how the grand picture of animal life, all the diversity and complexity we see, is built upon these incredibly sophisticated dynamic molecular and cellular foundations. And these foundations aren't static. They're constantly adapting through evolution over long timescales and responding dynamically to immediate needs within an animal's lifetime. Every living creature is really a testament to microscopic ingenuity.
So next time you see an animal move, maybe a bird taking flight, or even just notice a subtle change in its appearance, take a moment. Consider the silent, tireless work happening at the molecular level, all those proteins and membranes and signals orchestrating it all. What new insights will you uncover when you look closer? Thank you for being part of the Deep Dive family.